All right, hello everyone, and welcome to This Explainer. Today, we're jumping straight into a truly fascinating journey into the rules of heredity. We're going to shrink all the way down and explore the microscopic, three-dimensional world of genetics, or as it's beautifully called in Hindi, Anuvanshiki. So get ready because we are about to uncover the incredible, invisible codes that literally build every single living thing on Earth.
You know, it all starts with one fundamental mystery. Have you ever really stopped and wondered? Why do you look like your parents? आप अपने माता-पिता जैसे क्यों दिखते हैं? How exactly are physical traits, like your eye color, your height, or even the exact shape of your nose, passed down so flawlessly through generations? For centuries, humanity honestly had absolutely no idea. But today, we're going to map out exactly how that works step by step.
Okay, let's dive into this. Here's our roadmap for today's explainer. We'll hit Mendel's rules of inheritance, look at the dihybrid cross, explore Morgan, flies, and linkage, see how sex is determined, and finally, wrap up with understanding genetic disorders. Section 1. Mendel's Rules of Inheritance, the Foundation. To understand our genetic code, we've got to travel back to the 1860s. We're starting with the absolute bedrock of genetics here, Gregor Mendel and his meticulously cultivated pea plants. But before we look at Mendel's laws, let's quickly establish some vocabulary. Think of an allele simply as a flavor of a gene. Phenotype is what you actually see out in the real world. It's the physical expression of those genes. But the absolutely crucial concept here is dominance, or probuta. Visualize a vibrant purple flower gene pairing up with a white flower gene. Because of probuta, the purple allele completely hides or masks the recessive white one. So the resulting flower, it's completely purple, even though that white instruction is secretly hiding right there inside it. By tracking literally thousands of these pea plants, Mendel made a few core discoveries that completely changed science forever. He realized traits aren't just some blended soup of the parents. Actually, scratch that. It's more like they are unitary, discrete units. He formulated the law of dominance, which we just talked about. He also came up with the law of segregation, meaning when organisms create sperm or egg cells, the allele pairs split up fairly so each cell only carries one. And finally, through the law of independent assortment, he proved that a trait like seed color is passed down entirely separately from seed shape. Think of it like sorting laundry, right? The socks sort into their own pile completely independently of the shirts. Section 2. The dihybrid cross. Visualizing the math. Let's actually see what happens when we test that independent assortment idea by looking at two distinct traits at the exact same time.
And this brilliantly illustrates exactly what Mendel saw. This 4x4 Punnett square maps out a dihybrid cross for pea shape, so round versus wrinkled, and pea color, yellow versus green. The capital letters are our dominant traits, and the lowercase are recessive. When you cross two parents who are heterozygous for both traits, you map out all 16 possible combinations. It's kind of like looking at a 3D matrix of possible biological futures.
And here is where the math gets really cool. When you tally up the phenotypes from that matrix, you get this magical expected ratio, 9:3 to 3:1. 9 round in yellow, 3 round in green, 3 wrinkled in yellow, and just 1 purely recessive wrinkled in green. This mathematical perfection was Mendel's ultimate proof. It definitively proved that color and shape were handed down entirely independently of one another.
Section 3. Morgan, flies, and linkage, the exceptions. Now, biology is rarely that neat forever, right? We're now jumping into the 20th century to look at the exceptions to Mendel's perfect rules brought to light by a brilliant scientist named Thomas Hunt Morgan.
Morgan didn't use peas. He used Drosophila melanogaster, which is just the common fruit fly. Why? Well, because they're essentially the ultimate living data set. They thrive in a lab setting, they churn through a whole generation in just 2 weeks, and they mate to produce hundreds of offspring. Plus, you can really easily tell the males from the females under a microscope. Honestly, if you've ever left a ripe banana out on your kitchen counter for a day too long, you already know firsthand just how incredibly fast these flies multiply. Through observing these flies, Morgan noticed something kind of strange. Certain traits didn't sort independently at all. They mostly traveled together. He realized this was because the genes were physically stuck together on the very same chromosome, or in Hindi, Gunsothra. He called this linkage. However, they weren't glued perfectly, permanently. Sometimes during cell division, homologous chromosomes cross over and swap segments, creating brand new non-parental gene combinations. This genetic mixing is what we call recombination. Morgan tested this deeply by looking at how the alleles were arranged on the chromosome itself. Think of it like passengers on a bus.
If the two mutant alleles are sitting together on the very same chromosome, they're in the coupling or cis configuration. If they're split across two different buses, homologous chromosomes, they're in the repulsion or trans configuration. But here is the fascinating takeaway from this. Whether they started out on the same bus or different ones, in cis or trans, the frequency of recombination between those two specific genes remained exactly the same. The physical distance between the genes dictated the crossing over rate, and nothing else.
Now, what's really interesting about this slide is how these invisible microscopic chromosome maps actually translate into the real world. Just look at the diverse, beautiful kernels on these cobs of maize. Some are yellow, some are purple, some wrinkled, some smooth. This whole visual explosion of variety is the direct, tangible result of linkage, recombination, and crossing over happening right there at the microscopic level inside the plant's cells. Section 4. How sex is determined, beyond X and Y. Let's pivot to another amazing job that chromosomes do. We often think we know exactly how biological sex is determined, but nature is far more creative, and honestly, way more diverse than just the standard X and Y. We're obviously most familiar with the mammalian XY system on the left, where females have two X chromosomes and males have an X and a Y. Because females only produce X eggs, it's the male's sperm that ultimately determines the sex of the offspring. But nature has other methods. In the XO system, which you find in a lot of insects like grasshoppers, females have two X chromosomes, but males only have one. They're simply X0, lacking a second sex chromosome entirely. But then, you've got the ZW system, where the entire dynamic is completely flipped. Here, it's the female's ovum or egg that determines the sex of the offspring, not the sperm. Komodo dragons, butterflies, and even the chickens at your local farm all use this female heterogametic system, where males are ZZ and females are ZW. Remarkably, geneticists have found that the bird ZW system and the mammal XY system actually share absolutely no genes at all. They evolved entirely independently from completely different ancient chromosomes. How wild is that?
Section 5. Understanding genetic disorders, the reality. Now we transition to the final, and perhaps the most impactful, stage of our micro to macro narrative today. What exactly happens when there are mutations or errors in this intricate microscopic code of life?
So, the crucial point is this. Mendel's laws and Morgan's maps aren't just abstract academic puzzles for scientists to solve. When the rules of heredity malfunction, they cause very real clinical diseases in humans. By understanding these exact inheritance patterns, doctors and geneticists can trace, diagnose, and even predict medical conditions across whole families. We generally split these genetic realities into two clinical categories. First up are Mendelian disorders. These are caused by a mutation in a single gene, just like a single wrinkled P allele. Conditions like sickle cell anemia or hemophilia follow Mendel's dominant and recessive rules perfectly. On the other side, we have chromosomal disorders. Now, these aren't just tiny single-gene typos. They're massive structural abnormalities or changes in the overall number of chromosomes. Down syndrome, which is an extra copy of chromosome 21, or Turner syndrome, where a female has only one X chromosome instead of two, are prime examples of this.
And modern science has developed incredibly specific tools to detect both of these. For Mendelian single-gene disorders, we rely heavily on pedigree analysis. By mapping out a family tree over several generations, geneticists can see exactly how a recessive or dominant trait is hiding or expressing itself. For chromosomal disorders, we use karyotyping. This involves literally taking a photograph of the cell's chromosomes under a microscope to visually count them and check their physical structure, looking for the very abnormalities that Morgan's early fly theories first hinted at.
From monks carefully counting peas in a garden to scientists isolating fruit flies in a lab, we have come a very, very long way in decoding life. Today, we literally have the power to map the entire human genome. So it leaves us with a profound question to ponder. What secrets do your own Gunsutra, your own chromosomes, hold? And as our medical technology gets even sharper, how will mapping them change the future of your health? Thank you so much for exploring the microscopic world of genetics with me today on this explainer. Keep wondering, keep learning, and I'll see you next time.
Thank you so much for visiting my YouTube channel and my unofficial biology on Facebook page. Thank you.